Today we're looking at cell division gone wrong, and we've already seen what happens with the development of cancer when mitosis goes wrong. We've also focused on how hugely important meiosis is for halving the chromosome number and for bringing about genetic variation. So what happens when meiosis is faulty? Well, mistakes can occur in the DNA code. You've already looked at the process of DNA replication, and you've seen that although enzymes are present to try and correct mistakes, sometimes mutations can slip in. Sometimes also when the body has been exposed to some kind of nuclear or X radiation or an environmental toxin, the DNA can be damaged. And those mutations sometimes have very little effect on the organism, but sometimes they can be lethal and can actually cause the death of the organism. You also know, and you've learned this previously, that there are two types of mutations which exist. Gene mutations, sometimes also called point mutations, and chromosome mutations. Now, in terms of gene mutations, we've seen that a single nucleotide can change. The wrong nitrogenous base can be inserted, and that is going to bring about maybe a change in the whole process or in the whole protein itself. You're going to get a different protein expressed. If this here is our correct DNA strand and we delete one of those bases, we're going to get what we called a frame shift mutation. Everything will shift down one base, and you're going to see that you get what we call nonsense DNA being produced as a result. We're going to get something that is not correct. It does not code for the correct protein. That is a gene mutation. It can happen when a nitrogenous base is slipped in addition, when one is deleted. These frame shift mutations can be very small, and if it happens in your skin cell, you won't even notice it. But if it's going to happen in a cell that is undergoing meiosis and going to be producing sperm or eggs, then it can be crucial because it can be passed on to your offspring. So faulty meiosis can result in one single chromosome changing, or the content of one particular gene changing, or it can result in a huge change within the cell. And let's have a look at some of those huge changes, the bigger changes, not just one little nitrogenous base changing in one gene. You remember this process of crossing over, where we said during prophase 1, the homologs came to lie close together and they exchanged genetic material. Now, sometimes during that chiasmata, or the crossing over, a piece of a chromosome can actually break off, or it can get knotted and pulled off. It can get lost, completely lost, or it can reattach itself. And the problem is, if it doesn't reattach itself correctly, you're going to get quite a few genes involved in that particular mutation. Now, depending on the number or the particular type of genes that have been lost, the mutation could be mild or it could be severe. Here is a scheme showing a whole lot of different kinds of chromosome mutations. We can see if this was up here was our original chromosome. What happens in the case of deletion of a whole gene? We've taken out gene D, it's been deleted. Now, if gene D coded for something fairly small and insignificant, it will simply be a change in that small or insignificant protein. But if that gene D was for a hugely important enzyme, for example, in your digestive process, or an enzyme that controlled cellular respiration, then you're going to be in trouble if you don't have that gene. Another kind of chromosome mutation is insertion, or a duplication. So here we've got genes D and E, and they are duplicated. All right. Another kind of mutation is inversion. We've got genes D, E, and F, but now they run backwards along the chromosome. And that can be a problem when the enzymes are looking for gene D to make gene D's protein. It's not there. Instead, F is in its place. Another kind, and it's actually quite a common kind of mutation, chromosome mutation, is something called translocation. The genes of A, B, and C are there, but it's lost the rest of the chromosome, and instead of D, E, F, G, H, and I, those genes, it's somehow acquired P, Q, R, S, T, and U from a completely different chromosome. And so the genes have switched from one chromosome to another, and that results in huge problems for the organism because those lost genes cannot be found in order to make the proteins that those genes code for. So on this level, you can see at the biochemical level what happens with meiosis going wrong. And after the break, we'll focus on some examples of meiotic problems and how they turn out in the offspring.